For our next contestant from Team Faculty, we've got Dr. Xiaoyu Zhang, and he's going to help us take a closer look at chromatin modification in plant stem cells.
So just remember, you see the tallest tree today on my slide. So this is a tree that Alex sent me, and there are a few dots here that are actually, you can probably see organisms that would normally refer to as humans. So at the time of discovery, this was the tallest tree in the world and was about 370 feet tall. And since it was discovered, it grew a couple of feet taller still. It's probably around a couple thousand years old, and it's nowhere near the oldest tree discovered on Earth, which is 9,550 years old. Okay. So we don't work on trees exactly. We work on our abdominals. It's very similar to trees. The reason I'm telling you this is because really the whole above ground part of these plants are produced by a very small group of cells called the stem cells in the shoot apical meristem called the in the center of the shoot apical meristem. It's going to do two things. They keep on dividing and keep a small population as stem cells. So this is the maintenance part. And they also supply cells to other zones to differentiate into all the above ground tissues that you see. So based on gene expression differences, you can define a few different zones that are going to do very different things as the plant develops. Okay? So we work on chromatin modification. We came to realize that the modifications of chromatin probably plays a pretty important role in regulating the balance between this maintenance and the differentiation part. And the reason is, for example, this is a mutant that cannot trimethylate lysine 27 on histone H3. Okay, so what you're seeing here is actually a double mutant plant. Okay? The really good thing about this plant is it's immortal. So basically it lives forever. Okay? The bad thing is it's going to be looking like this forever. So it's a ball of undifferentiated stem cells. So the problem with this mutant is it cannot differentiate. Okay, so this is all stem cells cannot differentiate. Now this is another mutant that cannot modify another residue on histone H4. The problem with this mutant is, so normally the apical meristem is keep on dividing and providing cells to produce more flowers in this case. But this mutant is going to produce a few flowers and then quit and ends in the terminal flower. Okay? So developmentally speaking, the problem with that mutant is it cannot maintain the stem cell population. Differentiation, no problem. Maintenance, big problem. Okay? So what we really want to do is to figure out what happens in those tiny little cells. We need a few 100,000 of those. Each plant we can get a few dozen. So we are developing a high throughput tagging mechanism that we can use affinity purification, so bypass all the tedious work. You just have to grind up the plant and then do an experiment, and you can figure out what's happening in the cells as they're being maintained and differentiated into lateral organs. Okay? So this is being done in corn, so we're looking at specifically these cells to figure out how they're maintained, how they transition from one state to another. And hopefully in a year or two, when the corns are old enough or big enough, we can do some experiments.